Okay, now time is 10 and we want to start our seminar. Buzo and Anin, and welcome to the 80th installment of our 2020 Gilead Tibas virtual seminar series. My name is Gelar Raufi and I'm a PhD student in the Learning Design and Technology and Urban Sustainability at Wayne State University in Detroit.
And Anin, my name is Ian Smith and I'm a master's student in environmental science at the University of Windsor.
We are your hosts for today's seminar. I want to extend a big miguj to our guests for being here today.
To begin, we'd like to acknowledge that the University of Windsor and Wayne State University sits on the traditional territory of the Three Fires Confederacy of First Nations, Anishinaabeg, Peoria, Miamia, and Mississauga Nations.
We as a Windsor and Wayne State communities would like to move beyond acknowledgement, and we would like to not only give our respect to the traditional territory we are on, but also commit to learning and growing, starting with our partnerships and collaborations moving forward.
This is the first panel seminar in the new year. We are excited to announce that our next seminar will be a nonprofit panel focusing on topics of public participation in the lake and stream monitoring, a pathway to stewardship. Save the date, Friday, February 4th at 10 a.m. Stay tuned for more information to come.
Today, our guests will be discussing and taking questions related to the pandemic reflection on the radical interdisciplinary in the arts and science.
Allow us to explain how we arrived at today's panel specifically. Back in the summer and fall of 2020, the GLEAR T-RUS graduate students and faculty involved in the seminar planning brainstormed topics and featured speakers with traditional ecological knowledge and environmental sustainability being topics of interest and high priority. Over the past few months, we've engaged possible panel members, and the co-host team gathered with the panelists to listen and reflect on ideas for topics of interest within their work and communities.
We as a graduate student group have reflected on those conversations and prepared a list of questions and topics related to environment sustainability and the themes of intergenerational knowledge, language, in waste spaces, climate change, and engaging knowledge keepers and yacht. This panel extends on that discussions, and we are grateful that all of you have joined us today to listen, learn, and reflect. Most importantly, today's panel is focused on our graduate students and their training as environmental leaders of tomorrow, in partnership with our indigenous communities, knowledge and writers holders. Please join us in building a welcoming virtual community. We would like to ask everyone to turn their camera on and give a wave even for a minute.
So good to see everybody. Okay. So as part of the Zoom ground rules, we'll ask that everyone who is not a guest should be on mute and have their video off. Please confirm that if not, please mute and turn off the video now. Your chat facilitators today are Halima and Rory. Please send questions to them and they will pass them on to Galara and I. This talk will be recorded and posted online at a later date, and we would ask the technical team to initiate the recording now if it has not already been recorded. The format of the seminar will go as follows. Our guest will introduce herself and deliver an open discussion, which will last for about an hour. We are encouraged to stay for the last 30 minutes, which is focused on career mentorship, question and answer style discussion with Dr. Willet.
I will provide some brief context to our speaker, and then we will turn it over to Dr. Willet to provide us more information about herself and her work, as well as answer to the questions. Dr. Jennifer Willet is an artist, a Canadian researcher in the arts, science, and ecology, a professor in the School of the Creative Arts at the University of Windsor, and the director of Incubator Art Lab. She is a member of the College of New Scholars, Artists, and Scientists in the Royal Society of Canada. Willet is a leader in the Canadian bio-art community and works intermaterially as an artist and the curator in the field. In 2018, Willet opened a new state-of-the-art bio-art laboratory, and in 2020, is a storefront bio-art studio and the community engagement centered in the Windsor, Ontario, Canada.
Thank you. Thank you so much. I'm just going to share my screen. Well, first of all, I should give a brief introduction and say thank you. I would really like to thank Gleer and the University of Windsor, as well as Wayne State University, for hosting me in this event. I love talking to scientists. It's a really interesting group to communicate with, because a lot of my communications are with people in the arts and humanities. The other thing, I really enjoy talking with people at my home institution. I also get a little nervous. Last night I was a little nervous, putting in a couple extra slides, trying to impress you all, because this is my stomping ground and you guys are my immediate community. And I really, a lot of the work that I do, I talk about Windsor and about what is going on here. So it's just really a pleasure to be here, and I really appreciate you having me. So what I'm going to do is I'm going to share my screen, and I'm going to confirm that everyone can see my presentation. Are we good? Excellent. Okay.
Yep.
So this talk is called "This is BioArt's Moment." The pandemic has been a very strange time for me as an artist and as a mother. I simultaneously have spent most of the last 2 years of my life at home, chasing small children around while attempting to also do professional Zoom meetings. And at the same time, my genre of art production has become incredibly timely, prescient, and popular. BioArt is art that uses biology or biotechnology, and quite often the tools and technologies of science, in order to make contemporary artworks. So for 30 years under the name BioArt, but for millennia, artists have been using living materials and collaborating with living organisms towards producing art. In my genre, even before the pandemic, artists were using things such as cells, enzymes, viruses to make artworks. And so suddenly this field has really shot up in terms of its public prominence. So I have been incredibly busy while simultaneously working in my bedroom while doing laundry at the same time. So, let's see if I can get that to go. So I'm going to talk to you today as someone from the BioArt genre, which has become more public, about strategies that I can see from my own art practice for improving science communication and science entanglement with mainstream society moving forward. And I'm going to show you some of my artworks and how I attain it and what might roll out or be appropriate or interesting for people like yourselves in the sciences. So like BioArt, science now has become a hot public topic, and it's on the news every night, and people are debating and they're talking about the results and the da-da-da, and it's very exciting times. And it is really, there's become a moment where mainstream society is really looking to scientists as well as people within the medical industry to help us solve some of the greatest problems of our time. And that's very exciting. And I have to say the scientific community and the medical community has really risen to this challenge. I love this, title here for this paper on AstraZeneca. It says, "Making a vaccine in a year is like landing a human on the moon." While I was in my pajamas writing about BioArt over the last 2 years, scientists that I know were in the lab working as essentially frontline workers, trying to help us all solve and advance the problems that we were encountering. Particularly here at the University of Windsor, we became very well known for wastewater analysis and helping to develop protocols and procedures around that to have like an active analysis of contamination of COVID cases outside of medical data. So on the one hand, science and medicine has been incredibly championed and cheered on and really revalued at this moment. But at the same time, some of the challenges that the sciences have encountered have also become very public. And one of those challenges could definitely be science denial, right? Or people who misunderstand or misinterpret how scientific methodologies work, how data points or how data is interpreted, and how science as a methodology is continually reassessing itself. But the other thing that's really become very visible, not only in the scientific realm, but culturally, is that we're really seeing that there's a huge inequities in terms of how people are able to navigate this time, right? So we're really noticing that internationally, some countries have more access to vaccines than others. We're really noticing that different cultural groups are really struggling more than others, and that our society is incredibly classist, and that your class really determines your outcomes in this pandemic. I think also things like Black Lives Matter has become really like a cultural zeitgeist during this time, because people have had to stop and slow down and only see what are the inequities that different groups are facing. So science on the one hand has received a big hurrah and has really stepped in and offered a lot to our culture. But at the same time, there's a lot of naysayers, and also the facade of science as this like perfect knowledge form, as a form that does not discriminate, that is objective, is actually also cracking in public debate. And so for me, as an interdisciplinary artist and researcher, the cracks are where I go to. I get very excited when I see cracks, because that is where transformation can happen. So let me read you a little statement that I got about this I've been thinking about. So mainstream, so science is magnificent, but it is not objective. It is a cultural tradition, a Western hierarchical colonial tradition that set out to measure and frankly dominate the natural world. It does not recognize the value of other knowledge systems or knowledge outside of what can be measured. Scientific method is even sometimes harmful to its subjects of study, to organisms dissected in the lab, natural environments transformed by study, human bodies objectified in hospitals, and human populations or communities disadvantaged by the narrow stories that scientific data can tell us. So I want to simultaneously say that I have dedicated my life to engaging with science and biology and biotechnology. It is, for me, one of the highest forms of human knowledge, and creative and beautiful and wonderful, but it has these cracks, and these are things that we need to address. And science communication can fail us along similar lines, communicating only in the ways that science prefers, right? Rather than in the ways that benefit non-scientific communities. Scientific communication from what I'm here to propose today is scientific communication from a very different perspective, something that could transform society's relationship with science. So I would argue for things like improved lifelong science communication and science education. And I think we can all agree on that. I would argue for more informed public discourse and debate. I've been really excited over the last 5 years watching, for example, news coverage over the climate crisis becoming more and more engaged and enthralled and detailed. But the other thing that I'm here to argue for is for radical interdisciplinarity. And that radical interdisciplinarity, let me show you, I'll just actually move on through a couple slides and then we'll get to that. So these are some of the images that I found online last night while I was Googling science communication. And a lot of them are really like one-way forms of communication, where the scientist who has all of the knowledge communicates and shares that knowledge with a public, sometimes effectively, sometimes ineffectively. But the public is always like the receiver, and the public doesn't get arrows coming in and out of them. On this one over here, I even see society is like not even in the triangle of science communication. And so I pulled up some other images that reflect a more entangled, two-way communication vision of what science communication could look like moving forward. So if we look across the top here, you know, we have this idea that the information comes from the top of the heap, and that either goes down or it goes in this way, it goes in that way. And my argument would be rather that science needs to be one of the community members in conversation around these issues. That science communication is intrinsically 2 ways, but probably 16 ways. That we need, that communication starts with things like listening, right? Friendliness, open-mindedness, feedback, and nonverbal communication. So I would say that rather than just putting all of our efforts into creating better educational modules or better advertisements or better specialist discussions on television, that if science communication wants to take it up to the next level, they need to build entangled relationships with communities where science itself becomes transformed by those conversations. So what I'm arguing for is that we have to give up the disciplinary silos, that Science 2.0 needs to acknowledge its cultural bias and reimagine itself as a collaborative and entangled practice in collaboration with larger society. relying on methods and knowledges from non-Western traditions, as well as from the arts and humanities. Science needs to go beyond producing reproducible results and address the social cultural results of the data that it produces. So this is kind of my jam. I've been doing this for over 20 years. And so this is where the title of this lecture comes from. This is BioArt's moment, is that all of a sudden this very strange peripheral practice of these very unusual artists and philosophers and poets who have been engaging with science and a general public and creating moments for those two things to interact has all of a sudden become something that could be essential and could get us out of the dilemma that we find ourselves in. So I've been working for over 20 years in labs, but also in environmental research environments, creating immersive experiences, doing experience design, where I bring scientists and non-scientists together from all different backgrounds at all different age ranges to co-produce knowledge, hands-on knowledge of scientific processes. I highlighted here for you, I do a lot of this work outdoor in ecological environments, but I also do it in laboratory environments. So because a lot of you are students, I wanted to bring this back to the beginning. My first experience as a young artist going into places where I don't belong or working with scientists was during my undergraduate degree. I was at the University of Calgary and I wanted to do anatomical drawing. And I went to a professor and I negotiated access to the human anatomy lab at the University of Calgary in the School of Medicine. And I went in there once a week, maybe for a couple of years, and drew the cadavers. And of course, I learned a lot about anatomy and drawing. It really improved those skills. I frankly learned a lot about death. In our culture, where that aspect of the life cycle is really removed from our experience of being alive, and it's always seems sort of shocking when it happens or is like an ultimate failure. And it was really a powerful personal experience to also just spend a lot of time in a room with humans who have deceased. The other thing I would say about it is that I got to hang out with scientists and medical students. So I'd be in there drawing away and I'd hear some med students and they'd be saying things to each other like, don't cut the nerve. Don't cut the nerve. And I got to experience their experience learning how to navigate the interiority of the human body as scientists. So after that time, I wrote some notes of what I learned from this experience. And these became seminal. And I want you all to know this because even though you're at the beginning of your career now, some of the work that you do now will be with you for the rest of your life. So don't throw out your notebooks. So the notes from my experience of that time say, artistic education and research is not always understood or valued in the same way as scientific education and research in our society. Sometimes you will have to educate those from other disciplines about what you are doing and why it is important. And sometimes it is easier and more effective just not to tell everybody exactly what you are up to. Different disciplines have different community standards. Learn the language and standards of the cross-disciplinary community you want to participate in. Institutions rely on a form of professionalism, including moderate dress, good communication skills, and effective paperwork skills, in my experience. It will improve your chances of moving smoothly through institutional spaces if you adhere to these norms. Coming from the arts, I remember a lot of us had like blue hair and these types of things, and I found that that was a disadvantage if I wanted to hang out in the science or medical environments. Be respectful to people, communities, and institutions that differ from your own. You will learn a lot and make new friends and colleagues in unlikely places. And the last thing that has become my mantra is that it's often a good idea to go places where you don't belong. And now, even 25 years later, one of my assignments for my intermediate art class is I tell all the students that they have to go somewhere where they don't belong. They're not allowed to break the law, and they're not allowed to put themselves or anyone else in harm's way, but that's it. And the whole assignment is they have to go somewhere where they don't belong. They usually find out that they kind of belong there, or they don't. They learn some stuff and they come back and tell us that story. So now 25 years later, I run my own lab. This is a BSL level 2 art and biotechnology lab called Incubator Art Lab in the School of Creative Arts at the University of Windsor. It took a long time to get here. This lab is focused on the creative practices that engage with biotechnology. For many years, I worked in other scientists' lab and it was amazing and I learned a lot, but also I was always afraid of contaminating their projects. So this lab is designed specifically for our types of practices. So if someone wants to dance on the tabletops, that is okay with me. If someone wants to bring a baked good in and contaminate and put it in the agar, that's okay with me. And now the objectives for my Canada Research Chair are very similar to the ideas that came out of that very first experience working in the Human Anatomy Lab. So the primary objective of my Canada Research Chair in Art, Science, and Ecology is to devise research creation methods, artworks, texts, and technologies that serve to engage audience, species, and communities in collaboratively imagining possible biotech futures. So I want to bring all of these disparate groups together to co-produce the biotech future that we want to have, not allowing those decisions to be made in any one silo. The futures that I'm interested in imagining are feminist and include feminist and post-colonial perspectives, advanced digital and biotechnologies, historical, agricultural, and indigenous knowledges, interspecies collaboration, community engagement, sustainability, love, joy, gratitude, and the acknowledgement of suffering and death as inherent parts of the life cycle and of this type of research. So Incubator Art Lab opened in 2018. Before that, we had a little lab over in the Lavelle building, which I will not get into today. And it's a state-of-the-art facility that is rated a BSL level 2. It's a really hybrid space. We're like a generalist lab. So we do a little bit of everything, and then we buy lots of sort of like nimble, low expense equipment that can be pulled in and out depending on the projects that we want to do. It's currently, I received a CFI grant, Canadian Foundation of Innovation, where it's being wired right now as a theater. So it's going to become a performing arts venue that is also a science lab. So it has theatrical lighting, sound, video projection, video recording capabilities, so that we can do performances for live audiences, but also performances online. And I imagine those performances, things like BioArt opera, where maybe we have a traditional opera singer in the same space as a throat singer, participating in a cultural event, but we also are all cloning DNA or, you know, bioprinting or something along those lines. Just over the pandemic, we opened a public engagement studio, which is ridiculous. This is part of my research chair as well. I'm here right now, actually, and I'm in downtown Windsor, right across the street from the Capitol Theater. And it's a storefront artist studio. So I build all my big weird artworks here, but it also is a public engagement site where we can invite people to come in and do bioart workshops. You can see in the studio, we have a greenhouse. We're working on having that certified. It will be a BSL level 1 laboratory. So people can come in and do, like we can have school-age children come in here and do workshops, or people who are who do not have 3 days of health and safety training, they need to go in the other lab. And eventually that greenhouse, I'm right now transforming it into an artwork. So it itself will become this very bespoke object. I run my art practice like I run a lab. So 25 years later, I've worked so closely with scientists. I have contaminated their research, but they have also contaminated my art practice. So I have a team. This is a couple of years ago. We do new pictures every year. And we have artists on the team. We have scientists on the team. We have philosophers on the team. We have a nurse on the team. So I really bring together all these really interdisciplinary people who really thrive in the cracks, right? And together we work on my art practice. They work over years on different projects I'm developing. Together we develop community engagement projects and public events. We also help all of the students in the lab produce their own research projects. And in the arts, it's a little bit different than in the sciences. They author their own projects. I do not co-author their projects in any way, even if they're done under the lab. But the other thing that's unusual about in the arts is that usually people who assist on an artist project do not get a ton of public credit. So all the projects would be authored under my name alone. But I don't do that. I've learned from the sciences that it's important that that author list is really long and articulates all the different people who contributed to the project. Out of these spaces, I also run the BioArt, Contemporary Art, and the Life Sciences class. This is an undergraduate art class, but we also sign in students from all across campus. We just started live yesterday. Right now I think we have 6 scientists in the class, as well as students from various programs. And in this class, I teach the history of BioArt. We do hands-on BioArt projects. We do a lot of public events. We have an MFA program here in the School of Creative Arts, so I have master's level BioArt students. And these are usually people who have some previous experience with BioArt, will come to Windsor to really hone their skills and gain professional excellence in that field. An MFA is a terminal degree. So a lot of the students that come work with us in the lab are also like maybe adult students who have a lot more professional experience and are finishing up their education. We host a lot of events. This is the opening of the BioArt Lab in 2018, back when people used to have parties. And we did a big event called Feasting the Lab, where we had a big party and we did all the things in the lab that you're not allowed to do in a lab before it was certified. So we drank sangria shots in the lab. We danced barefoot in the lab. We had artists, we had scientists, we had Indigenous elders. We had everyone come and help us. We really feasted the lab the way you might feast a home or feast a new baby. And then we did all of this in one night. It was a big, like 500 or 600 people came down to the new SOCA building. And then at the end of the night, they gave me my paperwork. You can see it right here. And we stuck it on the door and we certified the lab, and everyone had to leave and put their shoes back on because we will never do that again. And I'm very interested in the bureaucratic processes around science, right? How does a lab become a lab? There's like this bureaucratic process that transforms what's possible in a space. We do a lot of art exhibits. I curate large-scale international art exhibits with big bio artists, but this one I also do stuff for the students. So this is our 5-year anniversary. In 2013, we had 10 of the best bio art projects that came out of the lab that were exhibited at the Ontario Science Centre. Then I took that class that year, a couple of classes that year up on a bus, and we did a bio art parade in collaboration with the Ontario Science Centre called the Eco-Nourri Parade. And we paraded all sorts of nighttime organisms through the streets of Toronto, organisms that we share our municipal ecologies with. So my overall practice is rooted in the notion that after all these years of experience, that biotechnology might be a technology of love. And I know this is a little unusual for the scientists. This work came from the startling realization that the time, care, and intimate knowledge and nudging it takes to sustain life in a hostile laboratory environment could be conceived of as a significant act of love. Scientists and bioartists are compelled to make substantial sacrifices in terms of personal relationships, health, and quality of life to maintain research experiments that can be so tedious and often counterproductive to human vitality. In this work, I'm not thinking about the unidirectional kind of love we might think a scientist could offer the organism of study. Instead, I'm thinking of a kind of love that entangles humans and non-humans in the mutually beneficial and mutually detrimental collaborative relationships. A kind of love that acknowledges suffering and even death. I wonder in what way, if any, we can speak of love or care afforded by the non-human organisms in the lab towards themselves, towards each other, towards the humans in the lab, and what kind of human relationships in the lab can also be guided by principles of love. So this piece serves as a mission statement for me. It's become the central locus of my art practice, but it's also become the central locus of how I built a lab and how I interact with the humans and the bureaucratic systems and the financial systems that support my research. So I'm going to show you a little bit of the type of artwork I do. So in addition to running a lab and doing outdoor ecological stuff, I also do a lot of fine arts exhibitions, right? So this is an exhibition that came out in 2020 called Baroque Biology. And it's intended to reproduce this idea that lab equipment and the aesthetics and design of lab equipment is not neutral. We think of it as neutral, but really it is very Western, it is very male, it is very digital, right? And so I decided to build lab equipment that would radically explode that idea. And so I made very feminine, beautiful, Baroque lab equipment. I painted all of the organisms on the lab equipment that might end up being used by the equipment. This is sort of going so far the other way. It's really interested in reimagining biotechnological research as more integrated with our domestic life, with play, with the stories that we tell each other. So in this, this is at the University of Waterloo, and you can see on the left here is a piece called the Great Lakes Algae Organ, which I will show you more of. On the right is another piece here called the Biosafety Puppet Playhouse. It's a functioning biosafety cabinet that we built that is also a Baroque Rococo puppet theater. And so the scientist or artist stands behind it and can do biotech protocols in the puppet house that can be seen by audience members through the plexiglass. This is a piece called the Great Lakes Algae Organ. And I have to say, Gleer really helped me out with this one. Whenever I'm working on a new project, I go talk to the scientists. So I had to come over to Gleer and learn more about like algae and what you got to do to keep it alive, and pumps. I didn't know about how much pressure you needed and the height ratio of how high you wanted the liquids to go. So this is a bicycle pulled portable laboratory that is also a street organ. And I exhibit it in museums and galleries, but I also take it to like fairs and parks, and it plays music. And the music is really interesting because on the one hand, it attracts audiences. So if I start playing the organ, like children start running towards me, they think there might be ice cream. So I usually have to have some kind of treat. But then they get there and they realize that they're going to learn about algae, and we have microscopes and we have little testing samples where you can taste the algae. The organ is also designed to meet the needs of the algae. So the algae requires light, it requires a nutrient solution, but it also requires agitation. And that's why we have this beautiful pump out the front of the organ. And then I often say that the music is not for the human audiences, it's for the algae to entertain them while they grow and proliferate. I also do a lot of lab work in the lab, and I just want to walk you through a piece called Baroque Biology Paper Theater. I got really interested in thinking about how microbes could maybe be performers for audiences, and how maybe they could be, that the microbes could be sharing their biological processes with us as humans, right? We often think about things like, we describe scientists as discovering something or inventing something, but maybe the microbes already had that information in their biological functioning, and they chose to share it with us or reveal it to us through these processes. So a project like this really starts off very small, where I'm doing little petri dishes and I'm taking some pictures with my cell phone. When I get an idea in my head and I get really clear in it, then I sort of mobilize the group, right? So this is some of our team members from a couple of years ago who you might recognize, and we're working on production of the Baroque Biology project. So you'll see in this environment, we're like making agar. We've learned how to dye the agar with different food coloring. We've learned how to put temporary tattoos on the agar. We've learned how to sculpt the agar. And then we're growing these sort of like little vignettes and petri dishes in the lab. I'm not a real scientist, I should say that. So what I tend to do is I find existing technologies, quite often like maybe consumer grade technologies, and then I hijack them for my projects. So this is an Amino Labs take home DNA and bioengineering kit, where you can create rainbow colors using bacteria that you genetically modify. So we made the rainbow, and then we reversed, found the recipe to create the agar, and then we used those rainbows as a palette, those rainbow bacteria as a palette. Then we started casting the agar, figuring out so these projects, very much like a scientific project, have many different sort of streams to them, and it takes a couple of years to figure out how to do it all properly. And then we started doing some of these initial tests where I would do within a single petri dish, little vignettes of like biological theater, essentially. And then these are some of the final prints. And what we did is we would have grown the petri dishes over a period of time, and then I placed them in the lab. These photographs are taken on the lab bench, and I just placed them on top of paper theater backgrounds. So these are traditional little puppet theater backgrounds, and we photographed them in the lab. I have also shown them live in galleries. The biosafety stuff is kind of complicated. I'm really good at it. So I usually get a biosafety officer from a local university to help me certify a space within the gallery, and then we go from there. So the other thing I want to talk to you about, while I'm thinking about science communication during the pandemic, I'm also an artist and I'm stuck at home, and I'm supposed to be running an international research center. So I did a lot of pandemic art over the last year or so. And I got thinking about how artists and people from the humanities can help our culture tell stories about this time, right? And that stories are often our ways of interpreting and internalizing and transforming our relationships to things like tragedies. So I started getting really involved in doing lots of little drawings and collages. I made a digital video with one of my assistants. During the first lockdown, I found out that I had hazmat suits in the basement. And so I pulled some of those hazmat suits out and I started drawing viruses on them and sticking beautiful stickers to them. And I came up with this piece that I call COVID suit. Although I have to say it has a nickname and it might be more appropriate. I call it the COVID suit mom. And the COVID suit became a way of making visible a lot of the anxieties and fears that we might have about contamination in the virus during this time. And then I started like going outside because we were just starting to go outside during the first summer. And I would like take my kids up and down the street for a walk wearing my suit. And the neighbors were like, oh, there goes Jennifer again. What's she up to now? I started crawling through the bushes in the park. And I booked a photographer to come join me one day. We shot some video footage. We're working on editing a video right now about some of the internal dialogue that someone might experience, and particularly a parent. I have to say that never has my role as a parent become so central to my research as while I was trying to conduct research while parenting at the same time. I did a lot of virtual work over the last year. And so I started doing all sorts of online workshops. Dr. Catherine Fabrio was a participant in one of those workshops. I created all sorts of characters in those spaces. We did a piece for Science Gallery Detroit where we built a paper puppet theater template that was also our lab, and then people could download it online and they would build our lab in their house, and then they would do little bioart experiments inside of the lab. Here's one I did in my backyard where I put weird food inside of my lab. The one at the bottom is a student of mine lit the lab on fire. I also did a collaborative project with IOTA Institute, which is a Canadian sort of intermedia arts organization. And we did a whole bioart online series where we did workshops online. It's called the BioArt Kitchen Hangout. So people kind of hung out and there were little hands-on things involved in a lot of those as well. We opened our public engagement facility during the pandemic, which is hilarious because I'm not allowed to let anybody inside right now. So we planned a virtual opening, which of course got canceled 3 times because we had more than 10 people together. And what we did is we shot this like 1990s MTV style video of me in a funny top hat running up and down the street of University Avenue, introducing myself to all the neighbors and being like, hi, I'm Jennifer. We have a science and art lab. And sort of learning more about what they do and what we do. And then we launched it live online. It was very funny and hilarious and weird. You'll notice that I really use humor, right? As a way to access new audiences. This is something I think science communication could really benefit from as well. I'm very open to self-deprecation. I think that when people are enjoying themselves, or if there's an element of whimsy that sort of breaks with the everyday reality of life, that they're more open to new thoughts and new ideas. I did a keynote lecture for the Global Community Bio Summit at MIT. And they called me up during the first lockdown and said, can you do a gratitude offering to the organisms in all of our labs? And so I negotiated with the University of Windsor. I went in while nobody else was there in the middle of the night, and I recorded a lecture that was an acknowledgement of love, and including pain and suffering for all of the organisms that were working in labs right now. I did this video where I sort of had this weird camera. I kept changing the angle of the camera. I'm really interested in upsetting how we use Zoom, for example. So I had a camera on the ceiling of the lab, and the very last shot of the piece, I turned on that camera and I laid down in this pile of organisms and I whispered like goodnight stories to all the organisms that we share our world with, including the COVID-19 virus. And then the last thing that I did recently is I flew to Slovenia last summer, which felt like going to outer space. I was terrified, to open an exhibition we've been planning called When Microbes Dream. And it's an exhibition that prioritizes the perspective of the microbes rather than of the humans in the lab. So all of these are sort of like allegories or stories of what maybe the microbes are thinking about or experiencing in the lab space. Now, I would like to really acknowledge that we have no way of knowing that. And that all this, the story I'm really telling is what human thinks microbes might be thinking or experiencing in the lab, but still just to invert that perspective is very interesting. On the left here, we have a spiral of algae going through tubes on a wall mural. On the right here, we have the biosafety puppet theater. A lot of my work focuses on the idea that the laboratory might be an ecology, right? And that the ecology of the lab is not separate from the planetary ecology that we are all engaged in. So this is an older work called Laboratory Ecologies, where I try to create this vision of a lab that is really multi-species. And again, one that deprioritizes the human scientist as the dominant organism working in the laboratory ecology. So the last thing I'd like to do is I'd just like to say that I'm not the only person out there doing this. First of all, there are thousands of bioartists and thousands of poets who work with science and all that type of thing. But the other thing I would like to say to the next generation of scientists that I'm talking to today is that scientists are also doing this type of work already. And so my argument for science communication is that not just that we have to change how we communicate or the types of adverts or the quality or the quantity, I think we need to change scientific methodologies so that they are more entangled, so that they are reciprocal, so that the audience you want to communicate with is also your collaborator. The audience you want to communicate also transforms the direction of your research. You know, we talked earlier about how a lot of disparity in how people are receiving access to care depending on what sort of different diverse groups they come from. If you want to invite Indigenous populations, for example, to be more receptive to scientific knowledge, you have to break through 150 years of science being used as a form of domination and control. And the way to do that is to work together, right? And to co-produce new types of science methodologies. So I just want to introduce you to 3 scientists who are doing this type of work, who I think are really cool and are role models for me. One is Dr. Andrew Pelling. Andrew Pelling runs Pelling Lab out of the University of Ottawa. I've known Andrew a long time. He's a very wacky guy. He has permanent artists on his payroll. And he makes science, but he also makes artwork and exhibits at galleries and museums all over the world. When he was doing his scientific training in California, he also studied under artists, okay? So it's not just that the non-scientists need more science education, the scientists need more arts and humanities education and learn how to engage with one another. So through his very unusual methodologies, which are very collaborative and very thinking outside of the box, he has developed new technologies where they are using vegetable substrates like asparagus. They're decellularizing them, filling them back with human cells, and they're doing clinical trials to use that as a way of creating replacement parts for human bodies. And it is really working. Another good example would be Dr. Katja Fabria, who's probably really embarrassed right now because she's turned her camera off. I have really enjoyed getting to know Dr. Fabria and the way that she runs the Healthy Headwaters Lab. This lab is doing water research. It's hyper local and it's in collaboration with different types of researchers. She has Indigenous researchers, she has artists, she has farmers, and they're all on the payroll and they are together solving these problems. And I think that that is a far more entangled way and a way where uptake of the type of knowledge they produce together could have real impacts. And the last person I'm thinking about is Dr. Max Leberon at Memorial University. And this is I don't know Dr. Leberon, but I want to go visit this lab, and maybe Catherine will come with me sometime, called the Civic Laboratory for Environmental Action Research. And this is a feminist, anti-colonial marine science laboratory, which means our methods foreground values of humility, equity, and good land relations. Dr. Leberon is of Métis descent. Dr. Leberon also has 2 degrees in fine arts. I put the CV down at the bottom here, a BFA and an MFA before doing a PhD in media culture and communication. So we want scientists who do not come from traditional scientific backgrounds, running labs, progressing research. So those would be my examples of people who are working actively in the sciences who have already uptaken some of the suggestions that I am making as an artist who works in the bioart genre. I think that is everything I have for you today.
Thank you very much. Thank you, Dr. Willett, for this engaging presentation. Before we enter the discussion period, we'll pause for a minute just in case some attendees need to leave. Okay. Then after that, we'd invite everyone to turn the cameras on and engage in discussion with our speaker. The portion that we'll be entering will be focusing on graduate students and early career experiences and questions. So we'll just give everyone a minute.
I wanted to mention on the side, I changed my lecture this morning, and then the changes must not have made it into the cloud. So there was one other little line of thought that I do a lot in my work that might be of more use to environmental research scientists. Is I do a lot of these like immersive projects where I take artists and scientists like camping. And if you bring people together over longer durations towards research outputs, the types of relationships that you can create are really durational and meaningful. And I think that what I'm arguing for here is that science communication will not be transformed unless we really transform those social relations between scientists and non-scientists. I'm actually supposed to be working on a project right now, but it's been delayed so many times with the pandemic, I'm not even sure if I'll do it anymore, called the invasive species fishing trip. And the original proposal was supposed to happen in 2019, and it's been delayed several times, where we're going to take artists and scientists fishing with different researchers at GLEAR. And we were going to go fishing with scientists, we were going to go fishing with indigenous fishing people, and we're also going to go fishing with subsistence fishers here along the Detroit River. And we were going to aim our discussion around invasive species and notions of invasiveness, and how sometimes the language of invasive species overlaps with our ideas and questions about human migration, and how we can maybe attempt to come to terms with the tensions that arise from that. So a lot of the work that I do is actually directly related to the work that you guys do. And I do bring artists camping and make weird artworks and unusual performances in those areas. And so for your own future research, I think that is a venue maybe where you could invite non-scientists to really engage in your work.
Shall we start with questions?
Yep, I think we're good. Okay, so as we're on the last 30 minutes of time, we would like to move on to the question and answer portion of the panel. Before we begin, are there any lingering thoughts or questions from the panelists that they would like to speak prior to answering questions from the audience? Otherwise, we can move on. Okay, so we'll move on to the question and answer discussion now. You want to start us off, Galara?
Yeah. So a question for the audience: what are your thoughts, feedback for the speaker? So how has social media helped or hindered your work? Is it a useful tool, or has it made interacting with the public in the detail more difficult?
I think social media can be a very valuable tool. But I think you have to be careful, because sometimes people have the sensation that if they've liked something or shared something on social media, then they have taken some action in response to a challenge. And I don't think that's the case. So we use social media a lot. We have a very public front. I think, look, if you want to engage a public, you have to go where they're at, right? So the public is on social media. So we do a lot of that. But what we try to do is we try to tie that social media engagement to real human interactions. So if you're going to do social media, you need great content, exciting, dynamic, weird, whimsical, unusual content, right? But then you also need to like chat people up, respond to their messages. Like, people are always a little surprised when they message our lab, and then I'll get on there and talk to them. And then I invite them to the lab, or I share a paper that we've done. So you really have to sort of go the extra mile to prevent that from just being like more meaningless. I will say, but it has really had some positive results for us. One of the guests here today is Lindsay, who is a graduate student who just started in our program, and she's studying remotely from Victoria, and we met each other through social media. So I'll say there have been real positive outcomes from it. The last thing I'll say about social media is it's a little harmful to the people who are doing the social media. And so sometimes I get really obsessed by it, and I'm liking, and I'm up all night like trying to figure things out. And I realize it sort of takes away from my regular lived experience. So sometimes I just allow it to come and go. We have a woman, Dominica Mediati, who I think is also here, who runs the social media for the lab in collaboration with me. I have my own social media, and you'll see sometimes we're highly active, and then sometimes we just take a break, or we don't, because we don't feel like it, because it can also be harmful to the person doing it.
Well said. Okay, so you've touched on this already, but do you have any advice for artists starting to engage in science or science communication? And likewise, where could scientists start engaging with art forms or artists?
Okay, I have lots of suggestions. So for artists, take the bioart class, read a scientific paper, go talk to a scientist. Like scientists and artists are a lot alike. We're actually like really smart and really into our thing, but we also kind of live in this little zone where you just do that one thing. So if someone from the outside says, hey, tell me more about your topic, I'm really interested in zebrafish, the scientist gets really excited and will talk to you. So I tell artists to introduce themselves to scientists all the time. One of my assignments is called Introduce Yourself to a Scientist, and you have to do like read a couple of their papers and then reach out to them and invite yourself to their lab to check it out. And 9 times out of 10, the scientist is so happy that someone is interested in their topic that they get the full treatment. So I would say the same thing for scientists. In fact, a lot of how I met some of the scientists at Clear was during my first year here. I had a big show at ArtSite, and I would say 3 or 4 of the kingpin top-notch scientists from Greer showed up at my art opening and said, we love art. And now we've had long-term collaborative relationships that came out of that. So as scientists, you should go to art events. You need to sign up for an art class. Do like one of those wine and paint things on a Saturday. Like it doesn't always have to be high-end stuff. Get a membership at the Art Gallery of Windsor. But I would also say that on artists, it is our duty to reach out across disciplines. So, you know, it's my duty to create events and then invite scientists, because it's a little scary going somewhere where you don't feel invited, right? So I think that I think you just have to, it's like making new friends, especially making new friends outside of school. It's a little awkward at first, but it's very easily achievable.
Great, thank you. And the next question is, how does someone train in practicing the conventional reductionist list, Western scientific method, go about breaking this mold and changing the status? Yes.
So I really recommend collaborating with people, or just inviting people into your zone. So some of my when I first started learning how to do this, I would find a scientist, and I would pretty much, any scientist who was nice and open to it, I would just do whatever they were doing. And I would go hang out in their lab. And maybe like go for a couple weeks or something, or a day, or a year. At the end of that interaction, the scientist would always come to me and say, you know, I knew we were going to share information with you, but I didn't realize how your questions and your presence would transform what we do in our lab. So one of the things that I would do if I was a scientist who wanted to be more open or learn more about how things can be done differently is I would invite specialists from other disciplines to come and just visit and hang out and have lunch. Maybe they come to your Friday meetings, like this meeting, right? And you're going to find they ask different questions. And those relationships, it's better if they're a little durational, but it can even happen on a short-term basis, because it's very hard. We're trained so acutely to like narrow our focus. It's hard for you to break out of that focus because you're so trained in it. And I'll say, as an artist, I'm also so trained in it that like I don't even see the assumptions that I'm making. And if you invite someone from the outside who has different assumptions, they'll say, oh, that's different than my assumptions. Why do you think that way? And then it opens up those conversations. We could also start an art science dating situation here at the university if anyone's interested in that. I have a lot of great artists who are really curious and who would love to come hang out in your labs.
Sounds fun. All right. I think this is one might be from the audience. After starting with the larger organisms, you said you started with drawing cadavers. How did you become drawn to microbes?
I like microbes because they smell bad. I'm really interested in things that are really beautiful but really gross. I love that feeling, that attraction repulsion moment where you're not sure if you're having fun or not. I like that feeling. So I tend to be attracted to organisms that do that. The other thing I really like about microbes is that I have an internal bioethic about what I'm cool with and what I'm not. And so like larger organisms or organisms with spines, I really hesitate to do that type of research because I'm not curing cancer. And I feel that I can get some of the same effects by working with organisms who maybe do not have the same level of sentience. But I will also say inversely that now after decades of working with microscopic collaborators, I've become far more open to maybe like what their experience of the world is. I sometimes think about the microbes and I think like if I stick my finger in the petri dish and crush Is that like a natural disaster? Are they having like news broadcasts about how a tsunami came out of the blue and killed off a third of them? Like, I wonder, you know, about what their experience of the interaction is.
Great. And the questions for the audience. What are your stories, experience you would like to share, or new thought that you have come from Dr. Willett's presentation?
You can type it in the chat, I believe.
Yep.
I'll offer a question because are your experiences with your art and when you do your show, is it different between Canada and US? Like just thinking about how T-Rest is Detroit and Michigan, just as the pandemic has rolled out differently. Yeah, just any thoughts there, just circling back with.
Yeah, so there's whole aspects of my career that we did not discuss today. And one of them is geographical nimbleness is really essential to the work that I want to do, because there are a lot of like legal, but also social barriers to different types of activities, right? And as an artist, I'm interested in exploring activities on a really wide scope. So I tend to work a lot internationally because it allows me to pursue different types of works in different environments. So I'll give you a good example. I used to teach at the University of Leiden in the Netherlands, and I could not do any research that involved, you know, even like they're very concerned about animal rights in the Netherlands. So that research would really be stalled in that environment. But the Netherlands have very open views around bodies and nudity. And so I did a lot of work because one of my goals is to see the human also as an organism in the laboratory environment. So I convinced all sorts of labs to let my friends and I come in and take our clothes off and then do biotech protocols naked or crawl inside of the hoods and stuff like that, because they're not offended by that. To them, that's a socially acceptable, just a body's just a body. Whereas I could never do that work in the US, ever. I can't do that work at the university I teach at because I teach there, and that would be a very unfortunate form of harassment. So I'm always sort of navigating the cultural differences between different environments. In Canada, I'll say bioart is huge because of the national funding agencies. Bioart is expensive, and it takes years, and it takes travel. And so SSHRC, the Social Science and Humanities Research Council of Canada, has become a massive funder of bioart. So has the Canada Council for the Arts. So there's a lot of bioart in Canada. In the US, there is also a lot of bioart, but it is different. It's commercial, it's incubator oriented. So there's a lot of bioart, but a lot more bio design, right? So people are making chairs out of mushrooms. And so there's quite often more of a commercial output in the work going on in the States.
Thank you. Okay, I got another one here. So since humans are destroying the ecological systems of the planet, driving a sixth mass extinction through destruction and contamination of biomes, as well as driving global climate change, etc., isn't the most important thing to do right now figuring out how to drive change in government policy? Art projects can be fun and personally enriching, and science has made our lives very comfortable, but at the end of the day, don't we need to be driving for political change as fast as possible?
Yes and no. Yes, I think that one of the best solutions to climate change is public policy. I think it doesn't, I'm like, my family calls me, like I'm a recycling crazy person. I cannot recycle our way out of this problem. Individual choices, but using reusable cups is not going to, we need massive public policies that drive industrial engagement with the planet. Number one thing. But how do you get people to vote for people who are going to put in those massive changes to our society? How do you get people to accept those changes and not protest? Or do you know what I mean? So very similar to the uptake of the vaccination process and the challenges that we've seen there, where on the one hand, it's actually really great. 85% of Canadians are vaccinated because they are getting the information. Some of them it took a little longer to persuade than others, but it's fine. How do you get that extra 15%? And so even though you think that when you're paying for art with public funds, that you're not paying for real outcomes, but what you don't understand is that the social aspect, the interhuman, interspecies aspect of being alive, of making decisions together, of caring about the future, of caring about our children, is driven by things like storytelling and the feeling. And if you only pay for public policy and more scientific data and you ignore people's feelings, it is not going to work.
Makes sense, thank you.
So that is the last question we will be able to get today. From all of us at both the Gilead and Tiros program, we would like to extend our thanks, Miguch, to Dr. Willet and to all who attend. We appreciate each individual sharing thoughtful answers to the questions and for being here today. Gilead Tiros team members, please stay behind.
Thank you so much, everyone.